Welcome back, my dear students. Today we are going to explore the molecular and chemical basis of muscle contraction, the hidden machinery that powers every movement of our body. To truly understand the molecular mechanism of muscle movement, muscle contraction, it's important to first have a clear idea of the ultrastructure of skeletal muscle, which we have discussed in the previous class. If we have not gone through that lesson yet, I strongly recommend watching the video first. It will give you the foundation you need and make today's topic much easier to grasp. Today we will explore how ATP, calcium ions, actin and myosin work together in the sliding filament theory to generate force. By the end of this class, you will clearly understand how tiny molecular interactions create power muscle contractions that keep us active every day. So what happens? What is the molecular mechanism of muscle contraction? When a nerve impulse or action potential arrives at the end of a neuron at the neuromuscular junction, then from the surrounding atmosphere calcium ion diffuse in within the neuron cell. Then the vesicle containing neurotransmitters such as acetylcholine fuse with the neurolemma or the neural cell membrane, and it will releases of neurotransmitter or acetylcholine into the neuromuscular junction. Then the neurotransmitter acetylcholine bind with the receptor present on the adjacent cell membrane of the muscle cell or the sarcolemma. These are the adjacent receptors of the sarcolemma, and the neurotransmitter will bind with the receptor. Then what happens? Then depolarization of sarcolemma occurs and a release of calcium 2 plus ion occurs from the sarcoplasmic reticulum, which are found just beside the T tubule. This is the T tubule, and just beside the T tubule there is sarcoplasmic reticulum where calcium 2 plus is deposited. They are released from the sarcoplasmic reticulum, and then the calcium 2 plus binds with the troponin protein of the thin filament of the myofibril, and this is the troponin, this is the myofibril, and this is the sarcomere. So first we have to know the structure what we have already discussed in my previous class, but in today's class we will again remind you the mechanism, the ultrastructure of the skeletal muscle. This is the unit of muscle, and what happens, this is the thick filament, the dark colored lines are the thick filament, and the red thinner lines are the thin filaments. So in between the thin filaments there is a Z line, hey, or zigzag line. These are the myofibrils. So in the middle of the thick filament there is a region which is known as M line, and the zone between this zone is the H zone, and this is the overlapping region where thick filament and thin filaments are aligned side by side. So these regions are A band or anisotropic band, and this thin filament region is the isotropic band or light band. This is the sarcomere. The region between the two successive Z line is the sarcomere, and in between the sarcomere there is A band. A band is consists of the thick filament, made up of thick filament, and in the middle of the thick filament there is M line. A band is the dark band and I band is the light band. So this thin filament is actually made up of three types of protein: actin protein, tropomyosin protein, and troponin protein. What happens? Actin protein, the globular actin proteins make a polymer, and it will form the fibrous actin or F actin, and then to this polymeric chain wound around each other and form a rope like structure, and the tropomyosin is positioned along the length of the actin fiber, but troponin is positioned somehow within gap. There are some gaps between the troponin component. So what happens in this is the thick region, thick filament, and it is made up of only one protein, that is myosin. So myosin proteins, these are the head of myosin, and they bind with the actin protein. And what happens, the length of the H band decreases. So the thin filament of two sides, they move towards each other, and they overlap with the thick filament, and thick filament, they sliding over each other, and this is the process of muscle contraction. What happens actually when calcium 2 plus ions binds with the troponin? Troponin, we know it is found with the thin filament. It is a thin filament protein. So there is this is actin protein, and this blue colored structure is the troponin protein, and just beneath the troponin there is tropomyosin. Beneath the tropomyosin, blue colored tropomyosin, there is a green colored troponin protein which binds with the calcium 2 plus ion. And this is the actin globules or globular actin. There is a site for of myosin head. The black region is the site for of myosin head. The myosin head actually have two parts. One region which has attachment with actin, and the second region have ATPase. So when calcium 2 plus binds with the troponin will pull the tropomyosin from the attachment. Tropomyosin actually positioned between the actin, positioned on the actin protein where myosin head binds. So if tropomyosin is there, myosin never can bind with the actin protein. So when calcium 2 plus ion binds with the troponin, then troponin will attach to the tropomyosin protein, and it will attract the tropomyosin protein, and it will remove the tropomyosin from its actual location, then the myosin head, so that the myosin head can bind with the actin protein. So this is when calcium 2 plus binds with troponin, tropomyosin will remove the troponin will remove the tropomyosin protein at the junction of actin and myosin protein. Then attachment occurs. Myosin head is attached with the actin. So what happens actually when myosin head attaches with the actin, then ATPase will be activated, and the ATP will be detached. It will form the, it will dephosphorylate, and it will form ADP and PI. Then attach utilizing this energy, the myosin head will attach with the actin protein, and then it will pull the actin proteins towards each other, and the sarcomere will be contracted, and it will bend and pull actin towards center. The actins will be moved towards the center. But when the muscle is relaxed, then action potential do not come, and ultimately calcium 2 plus will do not bind with the troponin. So tropomyosin will be placed in between the actin and myosin head, so that myosin head cannot bind with the actin protein, and the sarcomere will be not contracted. So now we see the excitation contraction coupling or EC coupling. The goal of the process is convert an action potential, whatever we have seen in earlier discussion, into a muscle contraction. So what are the steps in skeletal muscle? Motor neuron stimulation. Acetylcholine is a neurotransmitter released at the site of neuromuscular junction. Then end plate potential occurs, and action potential occurs in the sarcolemma or the muscle cell membrane. Then action potential spreads along the cell membrane of the muscle cell or sarcolemma and enters the T tubules. The T tubules contain voltage gated Ca2 plus channels or DHP receptors. Then depolarization causes conformational change in DHP receptors and opens Ca2 plus release channels or ryanodine receptor in the sarcoplasmic reticulum. Then calcium 2 plus diffuses into the sarcoplasm, binds with the troponin C subunit. Troponin C subunit is the subunit of the troponin protein which can bind with the calcium ion. So C for calcium, and it causes the contraction of the muscle unit or sarcomere. For relaxation or during relaxation, calcium 2 plus actively pumped into the sarcoplasmic reticulum. So what are the additional regulation? SR channels can also open via Ca2 plus induced Ca2 plus release and amplifying contraction. Then terminal cisterna of sarcoplasmic reticulum are closely associated with T tubules. It ensures rapid and coordinated calcium 2 plus release. So what happens when acetylcholine released from the action terminals binds to the receptor on the sarcolemma, and an action potential is generated and travels down the T tubule. These are the T tubules. So what happens? Calcium 2 plus is released from the sarcoplasmic reticulum in response to the change in voltage. So Ca2 plus binds with the troponin, cross bridges from form between actin and myosin protein. Then acetylcholine esterase removes acetylcholine from the synaptic cleft. Then calcium 2 plus is transported back into the sarcoplasmic reticulum, and tropomyosin binds active site on an actin, causing the cross bridge to detach. So what is the sliding filament theory? We have earlier noticed that the thin filaments move towards the center, and it will overlap with the thick filaments and slide over the thick filament. So this theory is called as a sliding filament theory. This only theory can explain the mechanism, the molecular and the chemical basis of muscle contraction. So what happens during states the sliding filament theory? Muscle contraction shortens the muscle fiber, which shortens myofibrils. Shortening occurs due to the sliding of thin or actin filaments over the thick filament, and not shortening of the filaments themselves. So key observation occur during contraction. A band stays the same length but moves closer together. I band shortens, only thin filament shorten, and H band shortens, only thick filament. Z disc move closer to each other, decreasing the sarcomere length. So what happens in the cross bridge cycle? Myosin head split ATP and become reoriented and energized. Then myosin head binds to actin, forming a cross bridge. This is the myosin head, attaches with the yellow colored actin, and the ATP will form the ADP and PI. Then ADP will be released out. So the myosin head rotate toward the center of the sarcomere. This is called as power stroke. As myosin heads bind ATP, the cross bridge detaches from the actin protein. So what is cross bridge? Myosin heads form a cross bridge with actin to generate contraction. Structure of myosin head: two globular heads per myosin molecule. We already know that ATP binding site or ATPase or ATP binding site is there. Actin binding site and ATP binding site. So function as myosin head as ATPase, it hydrolyze ATP to form ADP and PI. So myosin head split ATP and become reoriented and energized. Then myosin head binds to actin, forming cross bridge, and when myosin heads move towards center of the sarcomere, this is called as power stroke. As myosin heads again bind ATP, the cross bridge detaches from the actin. So ATP is split, myosin head phosphorylated, becomes cocked or stores energy. Then myosin heads binds actin, then inorganic phosphate is released. Power stroke pulls actin towards sarcomere center. When ADP is released, new ATP binds and myosin detaches from the actin. Then ATP split again, myosin head recocks, and then cycle repeats. Power stroke distance, that is approximately 6 nanometer per cross bridge, and multiple cycles required. Muscle can shorten up to 60% of resting length. Asynchronous action: cross bridges act like tug of war team, not in perfect synchrony. It ensures smooth muscle contraction. Fourth generation: more cross bridges engage when muscle load is higher. So what is the regulation of contraction? Muscle contraction is regulated by actin associated proteins. First one is the tropomyosin, which lies in the groove of actin helix and blocks the myosin binding site at rest. Second one is the troponin. It complex of three subunits. Troponin I inhibits tropomyosin binding. Troponin T, it binds with the tropomyosin, and troponin C, it binds with the calcium 2 plus. So during rest, tropomyosin blocks actin, so no contraction occurs, and calcium 2 plus when binds with troponin C, it moves tropomyosin, myosin then binds with actin, and power stroke occurs. What are these the role of calcium 2 plus in muscle contraction? Discovery: Sidney Ringer in 1883 firstly discovered that calcium 2 plus is essential for heartbeat. In relaxed muscle, low sarcoplasmic calcium, so tropomyosin blocks actin, so it cannot allow the attachment of myosin head with actin protein. So upon stimulation, sarcoplasmic calcium 2 plus rises, it binds troponin, and it will remove the tropomyosin from its position. Then actin will be exposed, and then contraction will start. So tropomyosin troponin complex acts as a switch. When calcium 2 plus is bound, it will promote contraction, and when calcium 2 plus is removed, it will promote relaxation. So what happens when electrical signal reception, then action potential propagation, then calcium release, then contraction initiation, then cross bridge formation, then power stroke, and after that is relaxation, and it will be followed by again another round of muscle contraction. Or so in today's class we discovered that how ATP, calcium ions, and actin and myosin work hard in hand to drive the sliding filament mechanism of muscle contraction. We saw that calcium unlocks the binding site, myosin heads pull actin filaments, and ATP provides the energy to repeat the cycle. Together, these molecular and chemical events explain how every simple movement from walking to breathing is powered. So always remember, behind every action lies a beautiful coordination of molecules at work. Stay curious, keep revising, and see you in the next class. Thank you.